MJ Hove presents COVID-19, Part 1, The Origin. This is Shi Zhengli, the Vice Director of the Wuhan Lab. Here she is conducting gain-of-function research in the Wuhan Lab. According to her resume, Shi Zhengli is a senior scientist with Wuhan Institute of Virology. According to her grant section, her funding in 2014 to study bat coronaviruses, she got money from NIH NIAID, which is Anthony Fauci's department. What was the result of the experiment that the United States funded? Published in the journal Nature Medicine in November 2015. A SARS-like cluster of circulating bat coronaviruses shows potential for human emergence. Shi Zhengli. They examined the disease potential of a SARS-like virus, which is currently circulating in Chinese horseshoe bats. Using the SARS-CoV reverse genetic system, we generated and characterized a chimeric virus expressing the spike of bat coronavirus in a mouse-adapted SARS-CoV backbone. SARS coronavirus is the human SARS virus from 2003. So they take the backbone, which is the basic structure of the virus, they implanted parts of a bat virus, and they generated a new chimeric virus, which is a hybrid virus. On the basis of these findings, we synthetically re-derived an infectious, full-length, recombinant virus and demonstrated robust viral replication. And here is an article warning us from the same month. This was in the journal Nature magazine. Engineered bat virus stirs debate over risky research. Lab-made coronavirus related to SARS can infect human cells. Now, bat viruses cannot infect humans, but what can infect humans is a lab-made coronavirus. And here's an editor's note added March of 2020, just in case we got suspicious. We are aware that this story is being used as the basis for unverified theories that the novel coronavirus causing COVID-19 was engineered. There is no evidence that this is true. Scientists believe that an animal is the most likely source of the coronavirus. An experiment that created a hybrid version of a bat coronavirus, one related to the virus that causes SARS, has triggered renewed debate over whether engineering lab variants of viruses with possible pandemic potential is worth the risk. In an article published in Nature Medicine on 9 November, scientists investigated a virus which is found in horseshoe bats in China. The researchers created a chimeric virus made up of a surface protein from the bat virus inserted into the backbone of the SARS virus. Simon Wayne Hobson, a virologist from the Pasteur Institute in Paris, said that researchers have created a novel virus that grows remarkably well in human cells. If the virus escaped, no one could predict the trajectory. You don't say. The argument is a rerun of the debate over whether to allow lab research that increases the virulence, ease of spread, or host range of dangerous pathogens, what is known as gain-of-function research. In October 2014, the US government imposed a moratorium on federal funding of such research. This study was already underway before the US moratorium began, and the US National Institutes of Health, the NIH, allowed it to proceed. The only impact of this work is the creation in a lab of a new non-natural risk, says Richard Ebright, a molecular biologist and biodefense expert at Rutgers University. Now, so there was a moratorium starting in 2014 that ended in 2017, December 19th, specifically 2017. Francis Collins, head of the NIH and Dr. Fauci's boss, issues this statement: NIH lifts funding pause on gain-of-function research. The funding pause was lifted in response to today's release of the framework for guiding funding decisions involving enhanced potential pandemic pathogens. The framework considers the scientific merits and the potential to create, transfer, or use enhanced potential pandemic pathogens. Do you understand what this is saying? It's saying, yes, they consider the scientific merits of such research, but more importantly, what they consider is the potential to create enhanced potential pandemic pathogens, transfer enhanced potential pandemic pathogens, or use enhanced potential pandemic pathogens. Deadly viruses with the potential to cause pandemics enhanced in a laboratory. What could that be used for besides biological warfare? And here, just a few weeks later, after the pause was lifted out of China, submission into GenBank, which is a repository for genetic sequencing of organisms, a direct submission submitted 5 January 2018 from the Institute of Military Medicine, Nanjing Command, Nanjing, China. They are submitting genetic information of a new virus that they collected in February 17, fully sequenced it, submitted it in January of 2018. Bat SARS-like coronavirus, Bat SL CoV ZC45. What is so special about Bat SL CoV ZC45? 2 years later, January 7th, 2020, in Nature magazine, the Wuhan coronavirus, now known as COVID-19, is compared against 16 other coronaviruses. Which do you think is most similar to COVID-19? Bat SL CoV ZC45, 95% match. Out of the 22 proteins, 18 of them match over 90%, and 2 of them match 100%. But don't believe that it's a bat virus that they made in a lab. Believe that it was from a wet market, from a pangolin or a raccoon dog. Don't trust your eyes or brain. Trust us, the people that enhance pandemic pathogens.